we have arrived at part 7 of Blood, page 644 again, where we didn't go over quite as yet the agranulocytes and the roles that they have to play, and we've been just sort of very non-specific about especially the lymphocytes. There are many kinds of lymphocytes, B cells, T cells of quite a variety, and so I'm pulling out this handout page right here, and we'll go over this for a moment. This is our handout having to do with everything you wanted to know about sort of specific immune response, but we're afraid to ask, so to speak. Here we have two kinds of immunity. I've given a dashed line. So here we have one scene one, or up here we have a situation where it's called cell-mediated immunity. And in this particular situation, we have a cell of interest here, a T cell. It's called a T cytotoxic, and it's specifically got a, I haven't really shown it very clearly here, but it has an immunoglobulin attached to the surface, and it identifies cells as it passes by. If it's an infected cell infected by a virus, or if it's a tumor cell in which it has become malignant in the cancerous sense, then that particular cell becomes the target for this T cell, and it lyses it with one of those sort of hollow spear type of arrangements where we attack the membrane and cause the membrane to leak out in many places, and that lyses the cell, destroys the cell. So that's a very specific kind of cell-mediated immunity. It's specific to virally infected cells and cancer cells. In graduate school in immunology, we calculated one time in a molecular biology seminar that with a really healthy immune system and an adequate number of these T cells, you could cure cancer about 11 times a second. Sometimes we're overwhelmed with much larger tumors than can be actually addressed with the immune system if it gets out of control. But we do have a fairly strong ability, and nature has made the arrangement for us to actually have cancer. There's gamma radiation coming through us at all times, and so DNA gets interrupted and mutated and sometimes promoted inappropriately. So the gene of interest here would be an oncogene, very necessary gene if you are a fetus or a embryo, because you have to grow quickly, but the oncogenes then need to turn off when the baby is born. So oncogenesis is something that we have a solution to, but we have to be not overwhelmed by it. Here we have a macrophage or a monocyte, and the situation here is that we have the monocyte has metabolized something. It has phagocytized, metabolized it, and then presented it on its surface in a way that can be recognized by other cell receptors. So we have a T helper cell here, another kind of T cell different from the one we were just looking at. So here we have a T helper. We always have name tags wherever we go. Class 1 or Class 2 major histocompatibility name tags. It's just a name tag. It's self. It says, hello, it's me. This guy is shaking hands with this guy. We have a B cell over here, another type of lymphocyte, also recognizing the antigen, also shaking hands with the various self-antigens, going, yes, it's me, yep, it's me. Right, so these are considered safe cells, and we don't normally, any of these powerful tools are not used against these cells because they're self. Once we have recognized, the T helper has recognized the foreign antigen, it becomes activated once this setup occurs right here with interleukin-1 activates T helper. Interleukin-2 will be released by T helper in two ways. In one way, it will self-activate the T helper further to the next step, and it will also, interleukin-2 will, and I will draw this in right here, it will go over to the T cytotoxic cell and activate that cell. I put a plus sign right there, and we have now activated T cytotoxic to do its work over here with virally infected cells or tumor cells. So, and this is interleukin 2, which does that. Okay? And so once we have a functional T helper that can activate the cell-mediated immunity, then we can also come over here to what's called the humoral immunity. And I'm using the old terms now, the B-cell growth factor, which is now an interleukin, and the B-cell differentiation factor, which is now a different interleukin, to activate the B-cell. This is a B-cell that can recognize the foreign antigen, and it's recognized as a special cell. It grows up to become a plasma cell or a memory cell, and what it's doing is it's producing antibodies. And once we get to that chapter on immunology, we'll learn much more about the individual immunoglobulins and the class switching and what's unique about them. So we at least should know that immunoglobulin G becomes present in the blood and is a response to an infection and stimulates a number of outcomes. Okay, so it also crosses the placenta so that it confers immunity to the unborn child. Okay, so that's that. That's why we have lymphocytes and a monocyte, and it gives you some ideas. Then we move on here to blood typing, and blood typing is an important feature. I'm just going to barely introduce this and go on to another part in order to explain it more thoroughly. But we can begin with the idea that there are red blood cells and that we all have a particular genetic disposition to the proteins that are present. I actually drew the proteins here on an AB blood type as an A and a B, if you can imagine little A's and little B's on the surface. Here we have a B blood type, and it has only the B proteins on the surface. Here we have a type A, and this type A only has A proteins on the surface. And here we have type O, which is by far the most common of all blood types, and it actually does not produce any of those proteins on its surface. It's free of surface proteins in that regard. There's another protein of interest that we might consider as the Rh factor, and we actually will consider it. But right now, just for the ABO blood types, it's important to consider it in this regard. And so what they're not showing here are they're only showing phenotypes, not genotypes. So basically, there's a couple different ways in which we can have this occur. If we have a type A situation, both the mother and the father could contribute to that. It could be just the mother who contributed, not the father, didn't contribute anything. He was a type O. Mom also could have been a type O, and then dad could be a type A. So that could be the genotypical contribution. Okay, and any of these will confer a phenotype of A. Right? There might be more or less proteins, but basically, let's do that. Show that it's an A and not a delta. Okay, so and the same thing with B. The B could be homozygous, that is to say both mom and dad contributed. It could be just mom contributed, dad did not. Could be mom did not and dad did contribute. So we have those options for type B. Okay, in the case of AB, it could be that mom provided the A and dad the B, or it could be the reverse of that. Okay, so those are the genotypes. In this case, there was nothing, both mom and dad had to be O. Okay, they did not contribute a protein, either of them. The only way that can occur is in the homozygous type O. Okay,